Yeah, hi everyone. I'm Shelley and my co-presenters are Kavya and Anjali for today. And so as I said before, we'll be presenting on the high-throughput virtual screening and chemical synthesis of efavirenz as non-nucleoside reverse transcriptase inhibitors, also known as NNRTIs, and how they enable the discovery of novel antivirals and therapeutics for HIV and AIDS.
So to start off, here's an outline of what we'll be talking about today. So first, what is HIV and what is AIDS? Well, HIV, also known as the human immunodeficiency virus, attacks the body's immune system, specifically the CD4 plus T cells or white blood cells that fight infection. When the CD4 plus T cell count in a person drops to below 200 cells per millimeter cubed in the blood, HIV progresses into its last stage, AIDS or acquired immunodeficiency syndrome. Currently, there's no widespread cure for HIV despite advancements in gene therapy, and thus treatment is the option for many. A popular form of treatment is antiretroviral or ART treatment, which puts an individual with HIV or AIDS on multiple medications to reduce the viral amount to an undetectable level. So NNRTIs and NRTIs are common types of these ART medications. NRTIs mimic nucleotides and integrate themselves into the viral DNA, while NNRTIs allosterically bind to the reverse transcriptase or RT enzyme, disturbing the enzyme's function and thus terminating viral replication. So here's an image on how HIV replicates itself using this reverse transcriptase enzyme. So it uses reverse transcription to integrate its RNA into the cell's DNA and then replicate itself. And thus reverse transcriptase HIV-RT is an ideal target for antiretroviral drug development. And so here are some FDA-approved NNRTIs, and our studies focus on efavirenz. Unfortunately, efavirenz was being phased out due to studies that correlate the usage of efavirenz with neurotoxic effects, as well as its poor resistance profile in relation to mutations in the HIV reverse transcriptase. But in other prior synthesis efforts, other compounds have been demonstrated to be neurotoxins, but the analogs of these compounds had reduced neurotoxic effects, making this a potential future for analogs of efavirenz.
Yeah, so now I'll be moving on to our in silico studies. Next slide, please. Yeah, so I'll be going briefly over like a brief outline of our in silico study. So we began by selecting the wild type HIV-RT protein from the protein bank RCSB and designing the library of our analogs. Then we used DFT or density functional theory to optimize the geometry and stabilize the library of analogs that we designed. And then we used Chimera to determine the binding pocket within the P66 subunit of our wild type HIV-RT. And then we docked the ligand into our protein using AutoDock VNAP. And the results were then analyzed to determine the binding affinities and interactions of the different ligands in the binding pocket using Chimera. So here's a closer look at our ligand docked within the wild type HIV-RT, as well as the various amino acid interactions it has. For example, a really key one is with tyrosine 181, 188, and 318. And here's our analog design. So what we did is, this is the R1 substituent, and we changed the chlorine on the original efavirenz with, this is the R1 substituent, and we changed the cyclopropyl on the efavirenz with cyclobutyl, cyclopentyl, cyclohexyl, phenyl, terbutyl, and methyl groups. And on the next slide, we changed the chlorine on the efavirenz with fluorine, methyl, and trifluoromethyl to get our final 112 analogs.
So here are the results for our wild type reverse transcriptase as shown in a heat map. So basically how you interpret this is warmer colors are better binding affinities. As you can see, our top analog was analog 2P with a cyclobutyl R2 substituent and a 6-trifluoromethyl R1 substituent. So the cyclobutyl group formed an additional hydrophobic contact with proline 95, which is one of those key interactions. And the 6-trifluoromethyl group brought closer and additional halogen pi interactions with phenylalanine 227 and tyrosine 188, respectively. After docking our analogs to wild type, we then docked it to common efavirenz-resistant clinical mutants, which consisted of 5 single mutants, so they only had 1 amino acid alteration, 4 double mutants, and 1 triple mutant. So here are the heat maps for our homology modeling, the efavirenz-resistant variants. And on the next 3 slides, I'll go into a more in-depth explanation of the docking. Okay, so this is how our ligands or analogs kind of look in the wild type or in the reverse transcriptase binding pocket. The yellow parts that you see in the diagrams are the key amino acids. The neon green is the mutated amino acid. The purple is our control, and the magenta ligand is our top analog. And as you can see, some mutants are more potent than others. For example, mutations at tyrosine 188, as that is a really important interaction that the ligand forms with the protein. And yeah, these are just more of those binding poses.
Yeah, so here are the top analogs from our homology modeling and our wild type. As seen generally, the top analogs consisted of the R2 trifluoromethyl fragment, as that particular fragment is able to maximize interactions. However, in general, we noted that trifluoromethyl analogs vary dramatically in performance, with poor binding affinities associated with 3rd and 4th position trifluoro fragments paired with large and bulky R1 substituents, and stronger binding affinities associated with 5th and 6th position R1 trifluoromethyl substituents, as well as smaller R1 fragments. So the former likely has to do with steric factors, and the latter is the ability to form bonds with important hydrophobic interactions. Yeah, and here is just more general trends. So from analyzing our docking, we drew a handful of conclusions. First is that our ligand mainly docked in 1 of 3 positions in the binding pocket, with the acyl fragment facing the aromatic amino acid cluster, glutamic acid 1138, and proline 236. And also we saw that high binding affinities were observed with analogs with 5th or 6th position fluorine and trifluoro fragments, combined with smaller acyl groups, specifically cyclopropyl, cyclobutyl, cyclopentyl, methyl. And we think, once again, this is because 5th and 6th position R2 substituents are able to maximize halogen pi interactions and hydrophobic interactions. And in addition, the fluorine and the methyl fragments did better than chlorine, which is what was originally on the efavirenz as an R2 substituent, because 1 fluorine has lower bond energy and thus an increased relative reactivity as compared to chlorine, and 2 methyl has an increased ability to form CH pi interactions. As for the smaller alkanes, they have highest binding affinity, most likely because of the larger ones having steric factors, and they're simply just too large for the pocket and thus resulted in diminished binding affinities. Yeah, so now I'll be moving on into like a new project that we might potentially start, and it's the synthesis of efavirenz.
So this is our proposed synthesis of efavirenz. It's safer and more efficient than the current Merck synthesis, which has 7 steps and uses a very dangerous and highly flammable reagent called N-butyllithium. So we'll be breaking down the synthesis into 3 steps. So the first step can be accomplished using 19F NMR to monitor the production of product. In addition, using quantitative 19F NMR, we can find the yield of the product as well. And so in this step, we can see methyl terbutyl ether and sodium acetate used as catalysts. The second step is a lot harder to accomplish without the use of that really dangerous N-butyllithium. And so in order to accomplish this, as you can see, a superstoichiometric asymmetric ligand and an alkyl lithium need to be used.
We propose using a metal and a chiral ligand instead, and we are currently working with zinc, ytterbium, indium triflate, as well as ligands such as S-binap, S-binol, and S-salin. And finally, for the last step, triphosgene in acetonitrile is used in a carbamate cyclization to obtain the final efavirenz compound. This step can also be monitored using Q19 NMR, as you can see using the top two photos. In addition, from the chart at the bottom, it can be seen that triphosgene is better than diphosgene in producing higher concentrations of efavirenz. So that is all we have for today. Thank you all for listening. We would like to thank our advisor, Edward, for helping us with this project, as well as ASDRP and the All Children Foundation for giving us this opportunity. Thank you, and we can take questions if anyone has any.